Hey folks, today we're going to be exploring the anatomy or structure of a skeletal muscle. We're going to be looking at both the gross anatomy and microscopic anatomy of a skeletal muscle. So just an overview of skeletal muscle. It is voluntary, it's striated, and it's multinucleated. Skeletal muscle extends and contracts and pulls on the bones to move the human body.
Skeletal muscle fibers are soft and fragile. They are bundled together by strong connective tissue that gives them the support and strength that they need. So the cream surrounding over the red is called the endomysium, which is the enclosing of each muscle fiber. Many muscle fibers come together to form a fascicle. The membrane of the fascicle is known as the paramecium. Many fascicles are surrounded by the epimysium, which connects to the tendon, which connects to the bone or cartilage. So we have the fiber, which is wrapped around by an endomysium. Many fibers come together to form a fascicle, and the fascicle is wrapped around by the paramecium, and many parameciums are surrounded by the epimysium, and the epimysium is connected to the tendon, which is connected to the bone.
Myofibrils are long organelles. They're thread-like proteins that push the nuclei aside in the skeletal muscle cells, as you can see right here. So they're like the microtubules of the cytoskeleton, and they fill the sacroplasm or the sarcoplasm, which is like the cytoplasm. Now, myofilaments are located in myofibrils. There's two types of myofilaments. There's actin and myosin. Myosin filaments are the thick filaments, or the dark, or the A band, as we can see right here. They have an H zone center. These red lines are the thick filaments. Actin is the thin filaments, or the light I bands. As you can see how it's structured right here, their center is the Z disc. It's a darker center. The H zone is a lighter center in the A band versus the I band. The M line is out here. Now, the thick filaments are made of myosin. They're bundled myosin. They're the entire length of the dark A band, and they have smooth ends, but they are studded with myosin heads. This helps with contractions. These myosin heads are cross bridges, and they can come together and link the thick and thin filaments together in contraction. Thin filaments are composed of actin, and they prevent the binding of myosin heads when needed. They're anchored to the Z disc, and they're adjacent to a sarcomere. Thin filaments do not extend into these sarcomeres, as you can see, and during contraction, these filaments overlap with myosin filaments as they reach towards the center of the H zone. In all, in summation, myofilaments give the myofibril a striated appearance. As you can see right here, it's an alternating dark versus light, and they're arranged in contracting units of sarcomeres. So their main job helps with contraction.
Now moving on, the myofibril, which is the actin and myosin filaments, are aligned in alternating patterns into contracting units, which are called sarcomeres. Okay, so within a sarcomere, there is the A band, which contains one myosin filament, the I band, which has two sections of actin filaments, the H zone, which has no actin filaments, just myosin, Z disc, which attaches and anchors actin filaments, and the M line, which is the center of the H zone.
Now, if you're a person who likes to remember tricks in mnemonic devices, we have one for you. So the A band is the dark band, and you can remember that because the second letter in dark is an A, so A to A band. It's the dark band with an A. And then for the I band, that is the light band, and the second letter in light is I, so the I's correlate to each other.
The sarcoplasmic reticulum is a specialized smoothie iron muscles. It surrounds the myofibril like a sleeve on a sweater. Its function is to store calcium and release it when the muscle fiber is stimulated to contract, so it provides the go signal for contraction. For other organelles, skeletal muscles have numerous mitochondria, which are the source of ATP because they need a lot of energy in order to contract. Transverse tubules are specializations of the plasma membrane that invaginate or cover the cell. They interact with the sarcoplasmic reticulum in order to shuttle calcium ions to the microfilaments.
We have just learned all about the anatomy of skeletal muscles. From a gross anatomy perspective, we learned that skeletal muscle fibers are first bound by an endomysium, then many fibers come together to form fascicles. Fascicles are wrapped by a paramecium, and then many parameciums are wrapped together with an epimecium, which connects to the tendon, which connects to your bones. Learned about skeletal muscle fibers, including actin and myosin, which are actin would be the thin filament and myosin would be the thick filament. These microfilaments help the muscle cell contract, and they give the skeletal muscles the striated or striped appearance due to their alternating bands. We also learned about sarcomeres, which are the units that actin and myosin are arranged into. We learned about A bands, I bands, H zones, Z discs, and M lines. We learned that muscle cells have a lot of mitochondria, which provides them ATP energy for contraction. We learned about skeletal muscles' specialized smooth ER called the sarcoplasmic reticulum, which releases calcium when a muscle fiber contracts and gives the go signal for the muscle. We also learned about transverse tubules that are specializations of the plasma membrane. They cover the cell and aid in the shuttling of calcium ions to the microfilaments. Thanks for watching, and we hope you learned something today.